Okay, so chapter 21 is about how populations evolve. So they call this population genetics, the study of how the gene pool of populations changes over time. So you might be asking yourself, what is a gene pool? So the definition, the gene pool is all the alleles in all members of a population. All right, so for instance, let's say you have a population and the species is diploid, meaning that there's two of every kind of gene. So each individual has two alleles for a particular gene, and they can be homozygous or heterozygous. So let's just do, for an example, let's do the population where there's 5 individuals, and let's say one individual is big R, big R. Say 2 individuals are big R, little r, and 2 individuals are little r, little r. All right, 2 individuals. All right, so that gives us our 5 individuals. And with this, to figure out our gene pool this is all the genes total. So if we have 5 individuals and they're diploid organisms, they have 2 of every kind of gene, 2 of every kind of allele, then do you see that you get a total of 10 alleles? There's 2 here, 2 here, 2 here, 2 here, and 2 here to give you a total of 10. So when we look at this, our gene pool, and we look at our total of 10 alleles, we have more little r's, like if you look here, than big R's. So our gene pool here, we have 1, 2, 3, 4. I'm going to do like a math problem here. 4 out of 10 is a big R, all right? And a little r is 1, 2, 3, 4, 5, 6 out of 10 is a little r. All right, so we would say if you divided this out, that's 0.4. So if you multiply by 100, that gives you, we'd say that 40% of our alleles are a big R, and this would be 0.6, and 60% of our alleles are a little r. All right, so that would be our gene pool for our population. So now let's go back up here to the definition of population genetics, and it says the study of how the gene pool of a population changes over time. So let's say, like right here, this is our original population and has 40 and 60, but let's say that something occurs in the environment that now, let's say, it's more beneficial to have a big R. All right, so what's going to happen to the big R over time if it's more beneficial? It's going to go up because the individuals that's the whole natural selection, Darwin's theory of natural selection. If you're more fit or have traits that are better suited for survival in the environment, that those traits, those individuals that have those traits will have kids and pass that gene on to their kids, and therefore that will go up. So I'm going to do a different color for the population. So what happens is if it changes over time, so let's say now we have 2 big R's and only 1 big R, little r, and 2 little R's. So now, can you stop this and do these calculations like we did for the original population? All right, so if you stopped it, you should get that the big R is 1, 2, 3, 4, 5 out of 10, or 0.5, or now 50% of the alleles. And then the little r would be 1, 2, 3, 4, 5 out of 10. 0.5 is 50%. So now we can see that the little r has gone down and the big R has gone up from 40 to 50. So we can put here, let's say that this is a change in environment that could cause that, where now it's beneficial to have the big R. All right, and so that's what it means by population. The gene pool of population changes over time. So therefore our frequency changes over time. And so we study that and look at populations, and that's how we can tell if a population is evolving or changing over time. So I want to point out that populations evolve, but individuals don't. Individuals don't evolve and change, but the frequencies in the population do. All right, and a population is a group of organisms, so that's that live in the same area at the same time. So if we look at the next bullet, it says and we're going to use this word fitness. The fitness of an organism refers to the contribution it makes to the gene pool of the next generation relative to the contributions of other members of the population. So in other words, a more fit organism survives and has viable. These viable means it's living and is able to have offspring itself. So like the babies can't be stillborn or not, so it has to be able to produce offspring that is able to live. So we would call that a more fit organism. And so organisms that usually do this are organisms that are better suited for their environment. So they have traits that help them to survive and reproduce in their environment. So remember that there is genetic variation in all populations. So therefore, that helps with survival, because if the environment changes, hopefully there's some individuals that have a trait that is able for those organisms and that population to keep on going and keep on reproducing. All right, so how we study the change in the gene pool of populations over time is through something that's called the Hardy-Weinberg theorem. So this is probably new to you, and we're going to learn it has some math to it. So the Hardy-Weinberg theorem is used to describe a population that is not evolving. So if we think about science, when you study something, you usually have a control group, something to compare it to. So in order to study a population evolving, we want to compare it to what's true about a population that isn't evolving. All right, so the frequencies of alleles and genes in a population's gene pool remain constant. So it'll always remain the same over the course of generations unless they are acted upon by forces other than Mendelian segregation, the recombination of alleles, other than just normal reproduction. Segregation, remember the alleles can line up in two different ways and segregate to different gametes and so on. So we can get some variation that way. Other than that, if anything, other changes occur, then that would cause a population to evolve. Now, when it says it's constant, what does that mean? So this would not be constant. From one generation to the next, we have a changing frequency here. Constant means that over time, this population would stay 40, 60, all right, big R, little r. So that's constant over time. So that we would consider that population not evolving if that's the case. This way, the example that I used here, from here to this over time, this population we would say is evolving. So the situation in which the allelic frequencies within a population are not changing is referred to as the Hardy-Weinberg equilibrium. So we would say that if this population stayed this way, generation year after year after year, generation after generation after generation, 40% big R's, 60% little r's, that population is not evolving, and we would say that that population is in Hardy-Weinberg equilibrium. If something is acting on it, like I used the example of big R's being more beneficial in the environment, if something is acting on it to cause that to change, the big R's and the little r's, then we would say that that population is not in Hardy-Weinberg equilibrium and that it is evolving. So what would be true, have to be true, for a population to be in Hardy-Weinberg equilibrium or not evolving? So what would be true for the R's, the big R to be 40% and the little r to be 60% over time? So we have a list of 5 things. So these conditions are required to keep gene frequencies constant. First is the population must be extremely large so that no genetic drift occurs. So we're gonna see in a little bit what genetic drift is. We're just gonna write it down right now. And so the larger the population is, the less likelihood that a gene is gonna disappear just by chance, all right? So that, so like a population of 5 here is not a very big population. So it could be, but when you have a really small population, that these two get the flu and end up dying, and it has nothing to do with them being little r, they just died. And since there's such a big percentage of the population, the percentages of big R and little r would change just due to chance. So the larger the population you have, the less likely that scenario would happen. And so if you had 2 little r individuals die out of a population of 1 million individuals, then it's not going to change the frequencies that much of big R and little r. Number 2, there must be no migration. So gene flow in the population. So no individuals can move in and no individuals can leave. All right, and so that's migration. So there's immigration and emigration, coming in, leaving. Because you can see that if you had a family that was all little r's come in and move into this original population, that's going to change the frequency of the little r's. There can be no mutations. So no new genes, and so the big R can't mutate to a little r or vice versa. So no mutations, because that can change the frequency. And remember, that means it's evolving. We also have to have random mating. Random mating must occur because, so therefore you can't choose who you're going to mate with based upon any traits that they have. So it has to be totally random, because if you're choosing based upon traits, then if you're looking for little r, then that little r is going to go up in frequency in the population. And so for that frequency to stay the same from generation to generation, that can't occur. So you have to have random mating. And the last thing is no natural selection, meaning that no natural selection can be taking place. So that means all the genes are equal, have equal survival rates. So it doesn't matter if you have a big R gene or a little r gene, everybody has an equal survival rate in the environment. All right? So how likely is it that any population at any time meets these 5 conditions? So imagine a population of an organism in the wild, and do you think that they meet any of these all of these 5 conditions all the time? What do you think? Probably not. All right, so usually you might meet one of them that maybe there's no mutation going on at that time, or at that particular time, one particular time, there's no migration, but usually there's natural selection. So to have all these 5 things occurring over a long period of time, because remember, we're talking about what happens to a population generation after generation after generation. Mutations can occur. Most of the time, populations do look for organisms look for specific things in their mate. Usually there's natural selection, that some traits are better suited for the environment than others. Not always is there a huge population, and so on and so forth. So most of the time, this is an ideal situation. In the wild, this doesn't really happen. So this would be like a controlled situation in like a laboratory or something like that. So that all these 5 conditions would be true. So let's look at how there's exceptions to this. So it says the population must be extremely large so that no genetic drift can occur. So let's talk about what genetic drift is. All right, so we have unpredictable chance fluctuations in the allelic frequencies of a small population. So I use the example up here. If you have a small population, if these two died of the flu, then you have a big change in your frequencies of big R and little r. The little r goes down big time, and the big R would go up if you are left with only 3 individuals. So this just gives you an example of that. So with genetic drift, there are two types of examples. So genetic drift, the first example is called the bottleneck effect. A sudden change in the environment, like a natural disaster, earthquake, or a fire, drastically reduces the population. So it doesn't kill everybody, but it kills off a lot of the population and leaves just a few survivors. So the few survivors may have a gene pool that no longer reflects the original population's gene pool. So let me give you an example. So if we turn this over, before we talk about this here, let's look at these flowers here. So here's your original population of flowers, and this shows incomplete dominance. So each gene, or each flower has 2 genes that determine the color. So this is supposed to be, I don't know why I didn't copy here, CR, where CR is red, and CW is white. So this has 2 red genes, so will be a red flower. 2 red genes, so all the darker ones on this picture here are red flowers. All right, can you see how these guys are kind of intermediate? So this would be, they have a CW gene, a red and a white gene. And so remember, this is incomplete dominance where they blend and you get the pink color, so those would be pink. And this one here is the one white with CW. All right, so what they did is they figured out the frequency of each one of these. Don't worry about these letters P and Q here. So the frequency of CR and CW is exactly what we did over here with figuring out the percentages. So if you counted, you would have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 for big R, or sorry, CRs, 14. And then for the CWs, you have 1, 2, 3, 4, 5, 6. All right, and so you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 individuals. So you have a total of 20 genes. So 14 out of 20 and 6 out of 20. All right, so if you divide 14 out of 20, you get 0.7. So that's the frequency. That's what of the CR gene. So that tells you that 70% of the population's genes are a CR. And here we do 6 divided by 20, that'll give you 0.3, and that tells you that 30% of the population's genes is a white gene. So that's your gene pool. So then let's say that some individuals are killed off, like we said over here with the bottleneck effect, that some individuals are killed off due to a fire. And it says only 5 of the 10 plants leave offspring. So if we look here, you see how these guys are highlighted in a white box? These are the survivors. These survivors, these are your 5, mate and repopulate. So only a small portion repopulates the, so these guys all die off. All right, they all die. And those guys are left to repopulate. So these guys might not be representative, have the 70% and 30%, 0.7 and 0.3. And so these guys repopulate. And now if we do the same thing and do the same math here, all right, and our frequency of CR, which is P, and CW, which is Q, changes. And so if we look over here, this is white, this is red, this is white. All right, this is white. This one is white, red, white. This one's the white. All right, so these guys are all pink. These guys are red. Whoops, and this is a red one. All right. and we have a few whites. All right, so if we count it up, 1, 2, and then 3, 4, 5, 6, 7, 8, 9, 10. So 10 out of the 20 alleles are going to be white, and 10 out of the 20 alleles, if you counted them up, will be red. All right, so therefore you get P and Q, or the frequency of the CRCW, as being 50% now. All right, and so we have seen how this changes the population. So then let's say another fire or an earthquake or something comes along and kills off the majority of these guys, and you only have 2 offspring left. So these are the 2, and they are left to repopulate the, notice that these guys are both red, so therefore they can only have red kids, and now you get 100% of your genes being CR, and only 0% being CW. All right, so the smaller the population is, the greater chance of fluctuation and change of your frequencies over time, just due to chance. It really has nothing to do with one gene being better than the other or anything like that. So that's why, in order for to get them to stay the same, that's why the population must be extremely large. Another example, the second example, is called the founder effect. A few individuals become isolated and establish a new population. So it's the same idea where you get a large population shrunk down to a small population. It's just the cause is different. And so this is where you have some individuals leave, let's say, the mainland and go to an island to establish a new population. So this gene pool is not reflective of the original population. So why they call it, they call this the founder effect, because let's say this is what happened with Darwin's finches. That doesn't look like South America, but if you have a bird's population on a mainland, and some of the birds go and populate a new island. All right, let's say these birds, there were some black ones, some gray ones, some white ones, all right, and let's say that a couple of black ones and only one gray one came over to the island. So therefore the frequency of your genes aren't the same as your original population on the mainland. And so the island frequency of your genes, the color genes, the black and the gray and white, are going to be different than on the mainland. All right, it's the same idea. So it's like we could say, for this, using this flower example, these guys in red, instead of these guys dying out, these guys and these guys surviving, the red ones go and populate the island. All right, and so then the island, when it's populated, is different frequency than the original mainland. All right, so it's just another reason why you might get a small population, and therefore the gene pool changed due to it being a small population. All right, and so I forgot to mention that they call this when this bottleneck effect, the reason why they call it that, when is it's random, like a earthquake or something like that, kills off organisms randomly. So if this is your original population with different colored marbles, so let's say we have black and white marbles in here, and the bottleneck goes skinnier towards the top, and you turn it over and shake a few marbles out, then you only get a small, random sampling of the population. That would be like the small random sampling of the survivors from a natural disaster. And so the surviving population may have a different frequency of your genes than your original population. So that's where that name bottleneck comes from. And so This is an example of the northern elephant seals in California. They were hunted nearly to extinction. So this is another thing besides a natural disaster. We can be the disaster as humans, where we hunted these seals to extinction so that there were only a few surviving members of the population. So the genetic diversity is not very great in that population anymore because of that bottleneck occurring. All right, so that's a real life example of that. So what can be the results of genetic drift? As I just said, you get a reduction of genetic variation within a given population, all right, for that reason, like the seals. And that can increase the differences between populations of the same species. So if you have a different population in a different area, you can have two different populations having two very different frequencies. Another exception to the Hardy-Weinberg equilibrium is something called heterozygote advantage. Individuals are heterozygous at a certain locus or location, which gives them a survival advantage. And what happens is this results in non-random mating. And so remember, in order to not evolve, you have to have non-random mating. All right. Sorry, you have to have random mating. I misspoke. And normally in most populations we have non-random mating. So the example I'm going to use here is sickle cell disease. So heterozygotes for sickle cell disease. This is a recessive disorder in humans. So you have 3 genotypes. Little r is you have sickle cell disease. So we've talked about this. These guys here you don't have. All right, so let's talk about the heterozygotes. Heterozygotes have both normal and sickle cells. And remember, the sickle cells, what's the difference between those? All right, sickle cells are shaped like a sickle, like this, and normal red blood cells are kind of biconcave. That's kind of a little indentation there. So this is a normal cell, this is a sickle cell. This, remember, interferes with carrying oxygen. O2, that says O2. The normal red blood cell doesn't, all right? So what they found is somebody who has little r, all the red blood cells are like this, all right? So therefore, they have side effects from that, and that creates the disease called sickle cell disease. People who are heterozygous have some sickle cell shaped red blood cells and some normal shaped. So, but yet they don't have the disease because they have enough normal shaped to get the oxygen to their cells and still be fine. Whereas big R, all their cells are normal and have the right shape, and so they have no issues whatsoever. What they found is that when you looked at the big R and the little r, near in the tropical places near the equator, they found that the sickle cell gene is in way higher concentration. So like where we live in North America, in the United States, the sickle cell gene is a low percentage, all right? It's not very high in our population. So what I'm talking about is the gene pool, all right? So little r, there's not very many little r people. And so therefore the little r is very small percentage in our gene pool. But as you go into tropical places, the little r for sickle cell goes way higher. And they found that there's a benefit from protection from another disease called malaria. And so people who are big R, little r have an advantage because the big R protects them. They don't have sickle cell disease. And the little r actually protects them from another disease called malaria. The sickle phenotype prevents the parasite from entering infected cells. So malaria is caused by a parasite that is carried and transmitted by mosquitoes. And so that's why in tropical areas where there's a lot of water and therefore a lot of mosquitoes, you have higher incidences of malaria. So therefore, people who are big R, little r, have a greater advantage because they have a resistance to malaria. Whereas if you're big R, while you don't have sickle cell disease, if you're bitten by a mosquito that has this parasite that causes malaria in it, you're going to get this disease. So this is in those tropical places, big R, little r is the better genotype to have. And so heterozygotes do not have the full-blown disease due to that dominant normal allele, and so they have an advantage. And this just shows you a map of the frequencies of the sickle cell gene is what this is showing you here. And so you can see that the darker colors have a higher frequency, and as you go north and south away from that equator, that the frequencies go down. All right, and so that's just showing you what I was just talking about there. All right, so let's relate this and go into a little bit more depth about this Hardy-Weinberg equilibrium stuff. So this is just a quick little review. So the Hardy-Weinberg equilibrium will only occur if. So if I were you, I would pause this and see if you can fill in these 5 things. These are the 5 things that if a population is not changing, what needs to be true. Go ahead and pause that and do this. So you should have that the population is extremely large. There's no migration, no mutations. Mating occurs randomly, and natural selection is not taking place. All right. So we're going to look at some equations. So the equation used to compare expected frequencies of alleles if the population is in Hardy-Weinberg equilibrium. All right, so if it's in equilibrium, the frequencies are going to stay the same over time. So that, you know, this population, if it's in equilibrium, the frequency of the big R is going to stay at 0.4 and the little r is going to stay at 0.6. All right, and these 5 things has to be true if those things are happening. And we're going to compare that from the actual frequencies of alleles if the population is not in equilibrium. And so this is called the Hardy-Weinberg theorem. All right, is we're going to look at comparing expected, what's going to happen if it's not evolving, to what actually happens. All right, and so consider a gene that exists in 2 allelic forms in a population. All right, let's say we're going to call the dominant frequency, all right, the big R has a frequency of, and we're going to call it P. All right, so this is the letters that we use. And the recessive allele, little r, has a frequency of Q. So whenever you see these letters, P and Q, P is always going to be referring to the dominant allele. It doesn't matter what letter you use, and Q is always the recessive allele. All right, and so before we do this part here, you have some room here, let's write some things down here. I'm going to go through an example of a population. So let's say, example, we have a population of flowers. And let's say the big R is red, little r is white, and let's say we know that 20 flowers are white, so that means they're little r, and let's say we know, I'm just going to give you this, let's say 160 of them are red, and we know that they're big R, little r, and let's say 320 of them are red and they're big R. So these guys look the same but have different genotypes. All right, so therefore that gives you 500 flowers total, all right, of red and white. All right, so let's look at, there's two things that we're going to look at. The first one is how to figure out the allele frequencies. So what's the frequency of each individual allele, the big R and the little r? All right, so that's the first thing we're going to look at. So we just said P is the frequency of the dominant allele. So P is, I'm just going to abbreviate frequency here, frequency of the big R. All right, so how do we figure that out? So since we know the number of individuals here, we can actually count them. All right, so therefore we have 320 big R individuals. So in each one of those big R individuals have two big R's. So we have 320 times 2. So we have 640 big R's from those individuals. But we also have the 160 red ones that have one big R. So 160 times 1 gives us 160. All right, and we add these together and we get a total of 800 divided by, we want a frequency, so that's kind of like we're doing exactly what we did here on the front page here, which is with more individuals. By how many total alleles? Well, how many total alleles are there? Well, there are 500 flowers. Each one of them have 2 alleles. So therefore, 500 times 2 gives you a total of 1,000 for the population, which means then that you get 0.8. All right, so 0.8 or 80% of the alleles in this population is a big R. We can do the same calculation for a Q. Q is the frequency of little r. All right, so we do the same thing. So we have 20 little r's. So 20, each one of those have 2, so we have 40 little r alleles based upon them. But then remember, you still have the 160 big R little r's. So 160 times 1 gives you 160. So we add these together and we get a total of 200 little r alleles. And out of our total of 1,000 alleles in the population, so that gives me a frequency of 0.2. So 80% of the alleles in this population are a big R, 20% are a little r. We call these allele frequencies. All right, so now notice here, that we call this P and Q. So our first equation is that P, these are the frequency of the alleles. When you add P and Q, what must they equal? P and Q, 0.8 and 0.2, always must equal to 1. Because that 1 is 100%. If we multiply by 100, it gives 100%. And so therefore, that makes sense that all of the alleles in the population should be a big R or a little r. So they should equal to 100%, these two things, the frequencies, all right? And they do in our particular case. So 0.8 plus 0.2 equals 1 in this particular example, all right? All right, so that's allele frequencies. The second thing we use with the Hardy-Weinberg equation or theorem is what is called the genotype frequencies. So we're going to fit this over here. So our genotype frequencies. All right, so we have 3 genotypes. So we have big R, and we have big R, little r, but we're gonna write big R, little r twice because there's 2 ways that you can inherit and be a big R, little r. You could have gotten the big R from mom and the little r from dad, or vice versa, the big R from dad and the little r from mom. So we can inherit this way or this way. And then lastly, we can have little r. So what we're gonna do is figure out mathematically how we can figure this out. So in our equation. So let's look at big R. All right, so how do we get to be a big R? If you remember the rule of multiplication, it's where you take 2 individual chances, the chances of one thing occurring and the chances of another thing occurring, and you multiply them together to get the chance of both of them occurring together. So what we do here is in this whole population, what's the chance that a big R is going to meet up with another big R to have a baby in this population that's big R, big R. Well, what you take is the chance, what's the frequency of a big R in the population? Well, 80% or 0.8. So you have a frequency of 0.8, 80% of all the genes. So if you imagine we put all the genes in a bucket and picked out a gene, you would have 0.8 or 80% chance of picking out a big R gene. All right, then you put that back in, and then you pick out again. To get a second big R, you'd also have 0.8 or 80% chance of picking out a second big R. So what's the chances of both of those that are going to occur together? You take the individual frequencies by themselves and multiply them together. So you have a 0.8 chance of picking out a big R out of the bucket or out of the population. And then we multiply that with the chance of picking out a second big R, which is also 0.8. So 0.8 times 0.8 is 0.64. All right, so that means that based upon these frequencies, we would expect 64% of the population to be big R, big R. Next one, so what I would do is pause this and see if you can do this, follow this logic to see if you can do it by yourself. So for big R, little r, frequency of big R is 0.8, little r is 0.2, so 0.8 times 0.2 is 0.16. But then remember, big R, little r can be inherited a second way, and so big R, or little r, 0.2, big R is 0.8, so we get 0.16. So therefore, really the chance of an individual being a big R, little r individual is 0.32. You add these together. So therefore, 32% of the individuals in the population, if it's in equilibrium, will be big R, little r. And lastly, little r, 0.2 times 0.2, that gives you 0.04. All right, and these guys should all equal 1. So 0.64 plus 0.32 plus 0.04 equals 1. And that makes sense because an individual in the population should be one of these three, big R, little r, or little r genotypes. So then how can we relate this and make an equation out of it? So let's think about this. So this was the big R individuals, right? So mathematically, how do we figure this out? Big R is P, right? So we took P times P. It's the same as P times P. It's the same as P squared. All right, see what I'm doing here? So then mathematically, see if you can figure out what you'd have to do to get your big R, little r's. We're using P's and Q's instead of the actual numbers. So what do we do here? For both of these cases, we multiply P times Q, all right, P times Q, but then we added them together or multiplied it by 2. So 2PQ, all right, multiply P and Q once, twice, 2PQ plus, and then here what do we do? Q times Q, so that's the same as Q squared equals 1. And this is our second, this was little r, this is our second Hardy-Weinberg equation. All right, so therefore you should be able to fill this in up here. So see if you can fill this in. Pause it and see if you can do that. So if we know the frequency of one of the alleles, we can calculate the frequency of the other allele. So we know that P plus Q equals 1. So we can solve for P or we can solve for Q by manipulating the equation. And the two are related by the equation for Hardy-Weinberg equilibrium, and that is this, so what we just came up with together. Whoops, that's supposed to be a plus. Q squared equals 1, where what is equal to the frequency of the homozygous dominant? That should be P squared. Heterozygous is 2PQ, and homozygous recessive is Q squared. All right, so let me give you an example problem here to just give you an example of what you're going to be doing on the some problems I'm going to be giving you in class. So example, all right, let's say this is the problem. 1 in 10,000 babies are born with a disorder called PKU. This stands for phenylketonuria. They have trouble digesting phenylalanine. This is a recessive disorder. All right, so everybody who has that is going to be little r, little r. All right, so my question to you is, what percent of the population is heterozygous? I'm gonna have you pause this and see if you can do that. Okay, and so using this, hopefully you tried it on your own, is you wanna start out with, so we're trying to find out big R, little r, but we can't tell somebody by looking at them if they're big or little r and are a carrier. So what you want to do is start with the people that you know their genotype for sure, and that's always the recessive. So 1 in 10,000 babies, so that's what you're given, 1 in 10,000 babies are little r, little r. They have the disease. What is that in Hardy-Weinberg equilibrium? Is that P, Q, P squared, 2PQ, or Q squared? That is Q squared. That's the frequency of the recessive genotype. That's a genotype frequency. So that's Q squared. If I know Q squared, can I find out Q? Absolutely. So I'm going to rearrange this. Q squared equals, and I'm going to put it in decimal instead of fraction format. So Q squared is this. All right, just move the decimal over. All right, and so when I figure out Q, then Q is going to be the square root of that, which is 0.01. I know Q. If I know Q, can I figure out P? Yes, I can. I can use P squared, or P plus Q equals 1. So I know I want to solve for P. So P is going to be 1 minus Q. So P is 1 minus 0.01. So P is going to be 0.99. All right, so then what do I want to know? Once I have Q, I can figure out P, then I can figure out anything else. I want to know heterozygous. Heterozygous is 2PQ. That's the equation for heterozygous. And so I just plug in my numbers. All right, so 2 times 0.99 times 0.01. All right, what am I going to get? I'm going to get 0.0198. And I'm asking you what percent of the population. So to get a percentage, I'm going to multiply by 100, and I get about 1.98% of the population is heterozygous. All right, and so you can use this if you know that the population is in Hardy-Weinberg equilibrium. And so this is an example of problems that we're going to work on in class and utilize. And what I want to point out is always look for Q. All right, you're always trying to get Q, and so never start out with individuals that have the dominant trait because they can be heterozygous, and so you might have some little r's in there, and so you can't start out with it. But you always start out with the people you know for sure, which is always going to be your recessive individual, and then you can figure out everything else from there. All right.